thanks a lot for giving me the opportunity to discuss our research topic with you. And and thank you, Dr. Bavle. And I got interacted with you a couple of times, and thank you so much for giving me this opportunity to be talking about our research today. Basically, today I'll be discussing about these nanocarrier systems targeted to cancer. Now, our current research objective in the lab is we are working on nanomedicines for bacterial keratitis. And for last 12 years, I'm working on cancer-targeted nanomedicines. So for ocular therapeutics, we just started very recently, and we also find that there are a lot of opportunities and possibilities to explore in infectious areas, especially with the nanoparticular drug delivery systems. And for cancer-targeted medicines, we started with the polymeric systems, and now we are going through some inorganic nanomaterials for anti-cancer drug delivery. So our main focus is to make combination therapy by multifunctional approach, where we combine chemotherapy with photodynamic therapy, or we also make the combination therapy with various drugs. And we also do the chemophotothermal therapy, where chemotherapy is combined with the photothermal application, where we irradiate the tumor and we get the molecules activated in such an extent that it will produce the heat which will kill selectively the cancer cells. So we have these few approaches in our nanomedicine research laboratory in Sciences-Pilani, Hyderabad campus. So for talking about the nanocarriers, we explore many different many different scaffolds. So we use the polymeric micelles. We also use dendrimers, human serum albumin, or other protein-based nanomaterials. And we also use the 2D functional polymers, which is graphene oxide, which gives the photothermal effect. For polymeric micelles, we have been working on this area for quite some time, and so far we have explored variety of polymers such as all amphiphilic polymers like polyethylene glycol polylactic acid. We also use the polyethylene glycol polycaprolactone. We use the polyethylene glycol PLGA polylactide co-glycolic acid. Several lipids also are used, so mainly all amphiphilic polymers. So we use the polyethylene glycol phosphatidylethanol so the conjugate of those two. We also incorporate vitamin A or vitamin E, the hydrophobic vitamins, to increase the power of amphiphilicity in the water-soluble polymers like polyethylene glycol or chitosan. We also use the hydroxypropyl methacrylamide systems. So that is basically highly hydrophobic, and it can conjugate various drug molecules in there. We also use the poloxamers, as I told you about, the poloxamers, as I told that we are working on bacterial keratitis. So poloxamer is something like thermo-responsive system. So we use these pluronic molecules to load the fluoroquinolone antibiotics for the treatment of bacterial keratitis. We also use several other systems, hydrophilic systems like chitosan. We also have the chitosan oligosaccharide lactate. We conjugate it with the hydrophilic molecules also to improve the mucoadhesivity. We also use the glycol chitosan, which is more hydrophilic compared to chitosan, and it actually addresses some of the disadvantages of using chitosan. One of them is its non-solubility in higher pH environment. So glycol chitosan will not have those kind of problems. So we use that also as a hydrophilic moiety to deliver the drugs through the cornea in case of bacterial keratitis. So now today's focus will majorly be the cancer targeted nanomedicines, as we have to as I will explore as I will explain eventually. So as I said that we use the combination therapy. So the several combinations which are tried is with doxorubicin, we tried oxaliplatin. Now both of them will have the DNA intercalating effect, but doxorubicin is topoisomerase 2 inhibitor, and oxaliplatin is also a very potent drug. But the problem with oxaliplatin is that it has non-specific binding because of its platinum structure. Now, when we convert this structure to a prodrug system, then the stability of oxaliplatin increases as well as the reactivity decreases. So we get the better safety profile in the prodrug system. So we went ahead and prepared some of the scaffolds with that. I will discuss about it later. So other scaffold that we are using is olaparib. We combined olaparib with oxaliplatin, and we prepared some delivery systems with that. And olaparib is highly hydrophobic material, and it has strong binding with the hydrophobic core. So we prepared some of these combination therapies. The basic objective for making these combination therapies is to obtain the robust therapeutic action. We also as I said that we also work on chemophotothermal therapy. So the light that we use is the laser source, which is approximately 808 nanometer. And then that is near infrared light, and we expose that to the tumor, and then we see that the temperature actually increases to up to 65 degrees centigrade, 45 to 60 degrees centigrade, and that actually causes the ablation of tumor. And along with this, there is a chemotherapy also, and which also helps to reduce the tumor. Now, what we use as a scaffold here is the graphene oxide basically. But then when we have the graphene oxide and we coat it with this our MPEG PLA molecules, which is amphiphilic molecule, and PEG is on the surface, so it actually helps preventing the opsonization by the reticuloendothelial systems, and we get and also addition of these amphiphilic polymers on the surface of the graphene oxide makes them spherical in nature. So spherical structures are easy to penetrate to the cell through the cell membrane, and also that will have the better drug delivery option. Okay, so and also there is so that another work that we are doing is the chemophotodynamic therapy, where we are combining chemotherapy with photodynamic therapy. Now, photodynamic therapy is different than all other therapies that I have talked about so far, because the photodynamic therapy requires the photosensitizer. This photosensitizer we are using here is chlorinese-6, and we are combining the chemotherapy with the photosensitizer, which is chlorinese-6. Now, again, the chlorinese-6 is also highly hydrophobic moiety, and it actually aggregates very rapidly, and the aggregation actually reduces its photosensitizing ability. Now, what basically it does and it is again dependent on light. but the light we have to irradiate the tumor with, that will activate the photosensitizer. Now different photosensitizers are in the market. Now we have to see the photosensitizers which are activated using the NIR light, because the NIR light is the one which has the deeper penetration. So here we are using chlorine-6, and chlorine-6 gets activated at 665 nanometer wavelength. Now we irradiate it, then it activates the photosensitizer, and the photosensitizer produces the singlet oxygen, and the singlet oxygen kills the cancer cells. So this reactive oxygen species generation increases quite a bit, and that actually is a killer for the cancer cells. Here in our studies, we have utilized the single formulation of chlorine-6, and also we have utilized the chlorine-6 along with the chemotherapeutic agent, which is oxaliplatin. So we have performed some of the studies with that. Again, as I said, that we also work with the bacterial keratitis, but I will not show you anything on that topic. However, we again make here as the nanomedicines, so but which is like, some little bit consistency, consistent gel form, which will adhere on the cornea, and then will slowly liberate the nanoparticles, which will have better penetration through the biofilms. Yeah, so as I said that for chemophotodynamic therapy, the chemotherapy that we have used is platinum drug, which is cisplatin or oxaliplatin, and the photosensitizer, which is chlorine-6. So you can see here that the chlorine-6 has a porphyrin moiety, and because of this constrained structure, this is highly hydrophobic, and whenever aggregated forms are present, that reduces their reactive oxygen species generation ability. And this cisplatin has non-specific binding because it is present in a PT2 oxidation state, but when we prepare the PT4 oxidation state molecules, by conjugation, their non-specific interaction decreases, so we get a stable version of the this drug. Now then we are coming to another so here I'm showing you that this is how we will get it. Here this platinum cisplatin, which is reacted with silver nitrate to make it hydroxyl group on the surface on it. So that actually is the first step to make the PT2 to PT4, and then it actually forms the DNA adduct. Okay, and then that causes the this captures the adjacent adenine and guanine and produces cytotoxic action. Now this OH is conjugatable, so we actually put the amphiphilic polymers in there, and can get the conjugation with the amphiphilic polymers to make it assemble into a drug delivery system. As I told you about the photosensitizer, when we put the light on, it goes to excited singlet state, and there by intersystem crossing, it goes to the excited state where it forms the reactive oxygen species. Also, it reacts with the biomolecules and form the reactive oxygen species, which is known as the type 1 reaction. The other one is known as the type 2 reaction. So these are like in a very briefly that what is going on. Now I will touch upon the pathogenesis of cancer. Why the cancer is so complicated, and I mean why there is no cure for cancer is the only thing that it's very difficult to target the cancer cells. And we can see that main thing, the problem is that the normal cell is not much different than the cancer cells. So if you see them, They are mostly similar, but if you see their biochemical, the signals, then you will see that there are multiple signals which are upregulated, multiple proteins which are downregulated, and a lot of things are going on in the tumor microenvironment, and it's a very complicated scenario, and where so many cells are involved for the cancer cell progression. Now we can see the tumor associated macrophages, the lymphatic vessels, blood vessels, then the neurons which are there, and all these different matrix materials, they all play a role in providing the cancer cells the ability for its progression. And if we see that, it's a very famous figure, which is from the Hallmark of Cancer, the review Hallmark of Cancer, which periodically gets published and talks about all the recent advancements in cancer. So this Hallmark actually tells us that what are the things which are happening with the cancer. So there are evading growth suppressors, avoiding immune dysfunctions, then abnormal proliferation, limitless replicative potential, genomic instability, and avoidance of apoptosis. So all these things are there in the cancer. So now how we can induce the apoptosis in cancer cells has considered to be a great therapy for cancer cells. But now it is possible to kill the cancer cells by many different means, but how we can target it to there, because most of the cancer related death happens due to the side effects of the chemotherapy, and the patient cannot survive the other organs which are getting affected by cancer. So that's the reason that targeted therapy for cancer is very much important to get developed. So now here I will just little discuss about the treatments of cancer. So you can see that we all know about all these treatment strategies now. As you know that the cancer has reached to households, and many near and dear ones of us are getting affected every day in cancer. And the diagnosis itself is a great challenge. Now when the diagnosis happened, then the problem with the medication or problem with the treatment strategies that it actually has enormous side effects, and which makes the morbidity very high, and of course the mortality in the cancer is still the highest. So I mean still the second highest. Now coming to the treatment options, chemotherapy, surgery, radiation therapy, immune therapy is also there, hormone therapy, stem cell therapy, the targeted therapies are also the approaches. Now coming to targeted therapy, we use with the nanomedicines, and we also know that the nanomedicines actually has some targeted actions which goes by the enhanced permeability and retention effect. And then there is also active targeting using specific ligands, which are specific to the tumor environment. And we also have the photodynamic therapy, and as well as the other stimuli that can be used are pH, light, or heat. Now coming to the cancer drug delivery, it is first of all, it's difficult to go to that tumor mass selectively. However, it has been found that if there is some the tumor vasculature is compromised, and obviously this happens to all the rapidly proliferating tumor microenvironment, so then that actually gives a way for the nanoparticles to enter the tumor environment. So that is called as the passive targeting, and all the nanocarriers show the passive targeting. However, the passive targeting is sometimes not enough because this passive targeting depends on many factors. So if this is the leakiness of the vasculatures, we have to know that the particles has to be smaller than these to go through this leakiness. That's the one thing. Secondly, the this leakiness is not same for all the cancers. In some cancers, the leakiness is small. In some cancers, the blood circulation populations are not there. Example, prostate cancer. Prostate cancer grows slowly because there is not that much blood infiltration. And what happens for this, that this EPR system doesn't work in that. Now, if you see the current trends, that there are a lot of nanomaterials in the United States which are getting approved day by day, and the number is actually increasing over the time. So it's not a very new data, but yeah, it is actually increasing over the time. A lot of drugs are getting clinically approved. Now, two very important drugs which are approved as nanomedicine in cancer therapy are Doxil, and that is the liposomal doxorubicin, and donoxazole, or maybe Abraxane, which is albumin-bound nanopaclitaxel, used for various indications like metastatic breast cancer, pancreatic cancer, then surgery, radiations, then advanced this non-small cell lung cancer. So all these indications, Abraxane is very useful. So what basically we have also done in our lab, we have used the human serum albumin as the nanoparticle system, because getting inspired by the therapeutic benefit that Abraxane is providing. So we actually used some of human serum albumin to load physically entrap multiple chemotherapeutic agents. Now, I will little bit discuss about the polymeric micelles and our work, very initial work on polymeric micelles. So what we basically prepared is a block copolymer that is MPEG PLA, and then we loaded the model and anti-cancer drug. Now, what is the benefit of micelles? Because they are very small size, which is maximum 50 nanometer. It should be less than that. Sometimes it can go little higher than the 50 nanometer also, depending on how big the hydrophobic part and how big is the hydrophilic corona you are putting into. It can have bigger size as well, but what the good thing about it, it's easy to prepare. Just thin film hydration in the laboratory setting can make the polymeric micelles. But if you want to make it in a bigger scale, we can have the dialysis option also to prepare the nanoparticles, and as it will assemble or undergo self-assembly, it will also take the drug along with it and make the stable nanoparticle, stable micellar core with the hydrophobic drug inside. There are different hydrophilic corona that can be used, and different hydrophobic core also can be used. Now, the hydrophilic corona, we can use polyethylene glycol. Of course, it's universally accepted. Chitosan can also be used. Various hyaluronic acid derivatives can also be used. Hydrophobic core, lot of hydrophobic cores can be explored, such as polypropylene oxide, because we already know about the fluoronic polymers which has PEO, PPO, PEO. So PPO is the polypropylene oxide which is hydrophobic, but polyethylene oxide is hydrophilic. Okay, so now coming to polylactic acid, polycaprolactone, polystyrenes. For the lipid, we use phospholipid, fatty acid, cholesterol, steroid molecules, or fat soluble vitamins like vitamin A or vitamin E. So these all are used. Now, in our initial studies, we have used the PEG PLA as the polymeric micelles. Now, what basically this will do? It will actually increase the solubility. So previously, if there is poor solubility of any material, it will cause the solubilization if you use the micelles. You can see here that in this, there is no solubility of curcumin. But if you put the micelles, if you add the micelles and check, then it completely solubilizes the curcumin. So you can see that how it is enhancing the solubility. Secondly, it will avoid the non-specific distribution. So as it will go through the neovasculature, and it will actually go through this leaky vasculatures and accumulate to the tumor. So it will produce and show the EPR effect. Also, another thing that if you have the liposomes and micelles, you will see that the micelles are penetrating better through these leaky vasculatures than liposomes because of their nano size, so which is actually beneficial in the in vivo applications. Now, also another good thing about these nanoparticles that they actually has low uptake and PGP efflux. So PGP, they can actually inhibit the PGP effluxing. Now, how do they do that? Basically, if you think about that whenever we have the free drug molecules, then they penetrate the cells by the diffusion, and then what happens that they cannot stay there because of these overexpressions of the P glycoproteins on the surface, which takes the drug or pumps the drug out from the intracellular environment. Now, that doesn't happen when we wrap the drug in the nanocarrier system, because that goes by the receptor mediated endocytosis process, and which doesn't accumulate in the periphery of the cells, because it goes through the receptor mediated endocytosis to lysosome by the endosomal pathway. So it goes through endosome. So first it forms the early endosome, then goes to late endosome, and from there it goes to the lysosome. So it doesn't get free in between. So it actually travels quite a bit in the intracellular condition, and so that, and then they liberate the drug. Therefore, there is less chance for that drug to be effluxed out, or the less chance for the drug to upregulate the PGP molecules on the surface. Okay, so nanoparticles actually has the inherent advantage for reducing the PGP efflux. We have used here this curcumin as a model drug, model hydrophobic drug, and it also has the property of anti-cancer. Now, the MPEG PLA that I told that we prepared by the ring opening polymerization reaction, and we took them and we prepared the thin film out of it, which is thin film hydration technique. And we added the PBS and to hydrate it. And after we added the PBS and hydrated it, we see everything went into the solution. And then we centrifuge it to get the micelles. So centrifuge has to be in a higher RPM to get the nanoparticles precipitated out. So this is the MPEG PLA polymer, which is prepared by the ring opening polymerization reaction in a sealed tube condition. And we use the MPEG here. OH acts as the initiator, and this DL lactide is used as the monomer, and this stannous octide is used as the catalyst. And we increase the temperature to 160 degrees centigrade to get these nanoparticles formed. So we actually do the typical studies with this. So we go for physicochemical characterization, which is must for any nanoparticle system that we will be using. So proton NMR. Then we also have to characterize that how the physical parameters of the nanoparticles are. So we find out the particle size, which is very interesting to see how the polydispersity index are. If the polydispersity index is above 0.5, it's not good. It's highly polydispersed with broad, peak. But always it's preferable to have it less than 0.2 polydispersity index, and the particle size also needs to be less than 150 to 200 nanometer to obtain the EPR effect. And the drug loading is another important parameter. So always when we work with the polymeric systems, we see that the drug loading is high compared to the liposomal system, where the drug loading is very low. And encapsulation efficiency has also proven to be high for curcumin in Pimpri PLA micelle. So it indicates that the curcumin has strong affinity with the polylactic acid, which is not causing the release very quickly. So we get the highest high encapsulation efficiency. Now we also have to see how the structure looks like. So we do the same analysis or TAME analysis. Then every time we prepare these novel amphiphilic polymers, we have to go through the critical micelle concentration determination, and we find that this is the pyrene method, the study done by the pyrene method. So we see the rapid sharp increase that indicates that the pyrene is getting incorporated. So the other studies that are done usually is the DSC, and also we perform the release study. So usually the release studies are performed at different pH conditions, because we need to understand that the lysosomal pH is less. So lysosomal pH is 4 or 4.5. So is the endosomal pH. So that drug release will not occur at 7.4, but if drug release occur at 7.4 but not at lower pH, then the material is not going to be releasing quite appropriately. So now we also perform the cellular cell studies. So always we perform the cell studies first with monolayers, and then we grow the cells in monolayers, and then we perform the experiments with the 3-dimensional spheroid structures. Now here for these curcumin loaded nanoparticles, we perform the cellular uptake study with the B16F10 cell line. So that, and also at different curcumin concentrations. So you can see that for B16F10 1 hour, the curcumin concentration increased, so from 50 to 100, and we see the higher internalization of the green signal. So the green signal is more here, less here. So that actually happened because we added more concentration over here. Now here also 4 hour we see a higher fluorescence, green fluorescence from curcumin, and here we put that as a geomean fluorescence, and we see that curcumin MPEG PLA at 50 microgram per ml has the highest uptake. So even same as here, that for 4 hour curcumin MPEG PLA at 100 microgram per ml has the highest uptake. So same study was performed in the human cell line also. So MDMB231 is a human triple negative breast cancer cell line, and we have found the similar findings for this cell line as well. Now this is the in vitro cytotoxicity study we performed with the MTT assay, and we find that almost all the tested points, there will be the curcumin MPEG PLA caused the higher cell viability or higher cytotoxicity compared to the free curcumin and MPEG PLA. We also perform the several active targeting approaches. Now, one of the approaches that I can show, I can show here, is the transferrin, the addition or modifying the nanoparticle with transferrin. Transferrin is a protein which around 77 kilodalton. Approximately, and then it actually it's a big molecule, so we can put it on the surface, and then that can cause the active targeting. And why the active targeting is required? Because the cancer cell has huge requirement of iron, and while taking the iron, they actually overexpress the transferrin receptors. Now this iron, iron actually has been a target for providing new strategies for cancer. So when we go do active targeting via these cancer this iron driven approach, there are a certain approach that also people follow, how they can quench the presence of iron in cancer, and then in tumor, so the cells will be deprived of iron and will go to death. Now, where here that again the transferrin modification is done on the polymeric micelles, we characterize, then we perform the cell-based study and three-dimensional spheroid structures. So the transferrin, incorporation of transferrin on the surface of the nanoparticles is not straightforward. So you have to use an amphiphilic polymer having the transferrin. So otherwise it will not stay like a nanoparticle. And so this anchoring potential is given to transferrin by conjugating it to the MPEG PLA, which is amphiphilic polymer, and will be embedded to the MPEG PLA polymeric micelles very easily. Now, what we did over here is we use the Traus reagents, and we made these we use the transferrin Traus reagents, and we make the MPEG PLA as terminally functionalized so that it can actually react with the sulfhydryl group with the MPEG PLA at the distal end. The micelles were also characterized with different approaches. Now, here we also have to the basic thing here that we want to achieve the targeting, and for that we should not compromise the particle size or PDI zeta potential drastically. So if you see that the particle size actually for non-targeted ones were 106, but which actually increased to 132, PDI was mostly similar. Zeta potential was towards the negative side because of the presence of that protein which is negatively charged. And then we also see that encapsulation efficiency little bit decreased, but not significant, and drug loading also decreased because of the presence of this sterically hindered transferrin. We also performed a cellular uptake study. As you see that this is our main, main study that actually enhancing the cellular uptake. So if you see that this curcumin MPEG PLA, which will have moderate cellular uptake, and we see that transferrin curcumin MPEG PLA, which is high, very high cellular uptake. So compared to this, we have a huge uptake that indicates that the targeting action is established. So here we have used, in this case, we have used the HeLa cell, which is cervical cancer cell. Here we have used the HepG2 cell, which is a liver cancer cell. The liver cancer cell is usually used because the transferrin is overexpressed in the, transferrin receptors are overexpressed in liver cancer cell because there is a lot of requirement for iron in liver cancer cells. Now, we also do some studies with the normal cells, and we see that the normal cells do not take up these formulations at all. So we see that there is no, green signal indicating that these tight junctions of the normal cells are not allowing any nanomedicines to enter in their systems. Now, we also perform some of these, how the nanoparticles are getting internalized. So we, I told that there will be receptor mediated endocytosis process. So we have the different other approaches like clathrin-mediated endocytosis or caveolin-mediated endocytosis, or it is macropinocytosis or not. So we have taken that and we inhibited different mechanisms, we inhibited different endocytosis process, and we find out that how the nanoparticles are getting internalized. I previously showed you the cytotoxicity data, and we see that the nanoformulations are always effective. In this case, the free curcumin, it's combined, the controls are free curcumin and curcumin loaded in MPEG PLA. So here, whatever action that we see for the curcumin transfer in MPEG PLA is being compared with the curcumin MPEG PLA, and you see that this only presence of transferrin makes the nanoparticle, the difference in activity between the blue signal and the green signal in the nanoparticles. We also performed the spheroid study. Spheroids are something like that, mimic the three-dimensional environment. So 3D environment we can create in the laboratory setup, where we basically use the cells grow in sphere form. And so when the cells grow in sphere form, then there are a lot of markers they overexpress, like adhesion molecules, because when we are growing them in monolayers, this is not a condition that the tumor actually experience. So it's like they grow in a group. So we actually provide this environment, even though we don't provide them with all other tumor microenvironment associated cells like macrophages or neutrophils or any other immune molecules. We don't do that, but at least we provide an environment where they grow in cluster form, mimicking the actual tumor environment. So here this is good model to screen the anti-cancer drugs, and we see that our nanoparticles, like which is actively targeted, is basically better in reducing the tumor volumes compared to the normal MPEG PLA loaded with curcumin. The spheroid study also, we performed that how the nanoparticles are getting internalized, and we see that the targeting action is predominant, that we see that a better fluorescence signal for this, indicating that these nanoparticles are getting internalized to a higher extent compared to just curcumin MPEG PLA, which is over here. So this is the 4-hour study. This is the 4-hour study. So we can compare this with this. Now, this is the 1-hour study, and this is 4-hour study. So 4-hour, it is internalizing much more compared to 1 hour. And then we also check their live dead cell probabilities, like that they are actually when we treated them with the propydium iodide, which shows the dead cell population, and calcium blue, which shows the viable cell population. So we see that basically in control there is no red cell population. So that means they are completely they are not undergoing apoptosis, or they are not experiencing cell death. But when we see that the transferrin labeled formulation has the highest cell death compared to these formulations, so it actually indicates that the targeting action actually killing the cancer cells. So we also do apoptosis studies with the annexin V acids, where this annexin V binds with the apoptotic marker phosphatidylserine, and then that actually causes the upregulation that we can find out by the FACT study. So we actually highlight this quadrant 3. If you can see that transferrin has a 34.3, but which is 25 for the free curcumin, and curcumin MPEG PLA, which is 29, little bit more, but this one has the huge apoptosis going on in the cancer cells. We also have several other studies that we have increased the drug loadability by improving their hydrophobic nature. The way to increase the hydrophobic nature is we had the PLA where we conjugated the cholesterol. So then after that we put the curcumin in that, because this also forms the micelles. So this so it's not, yeah, it's not just cholesterol. We also can use several other hydrophobic materials, as I told that we can also use some of the other hydrophobic moieties. So vitamin E or vitamin A is one of the options. So we go for vitamin E. Then we also put the oleanolic acid, another hydrophobic material, and that actually causes the synergistic action. We have the albumin systems, as I told you, that it's very emerging to use the albumin because of its biocompatibility, biodegradability, especially it's non-immunogenic, and also it has the ability to target the cancer cells. So those few advantages make the albumin a very good nanocarrier for cancer targeted drug delivery. So we are currently exploring that system as well, and we can see here that we actually use some of these systems, and in order to deliver the olaparib that I showed in the beginning, that we are also working with olaparib, and you can see that when we have the human serum albumin nanoparticles, then it actually compared to olaparib, it actually reduces the tumor to a greater extent that we see by our in vivo live animal imaging. We also see that when we use this kind of human serum albumin system, then it actually prevents the lung metastasis as well of the breast cancer cells. So we had the breast cancer, the tumor, the triple negative breast cancer in animals, and then after that surgery is done, then we allow them to survive, and eventually they develop the lung metastasis, which is very high in case of control. And with the lung metastasis, the lung become bigger in size, and with lot of nodules develops, and we can see here the red signal, but that is not found in the olaparib loaded human serum albumin. So that means that it is actually preventing the metastasis in lungs quite drastically. So we also have some of the next generation nanomedicines, which is coming up. As I told you, that there are a lot of possibilities for those, including photodynamic therapy. It's a simple molecule, but it actually has a lot of potential than chemotherapy, but because it actually selectively targets, because we can put the radiation, we put the light, and then we can activate it selectively. And photothermal therapy has huge potential because the temperature increases in such a way that we get the cell killing at 60 degrees centigrade. The cell actually automatically gets killed and tumor volume gets reduced drastically. Then combination therapy, definitely it's in the market, but not with the nanomedicines, but with the conventional therapy. We need to introduce the nanomedicines for the targeted anti-cancer action. Then combination of photothermal photodynamic, combination of chemodynamic photodynamic. So all these different strategies need to be developed. We also, so as I was mentioning to you, that why the HSA is better, because it actually targets the endothelial cells with these overexpressed glycoprotein 60 or another glycoprotein, which is SPARC. This SPARC is also overexpressed in the tumor. So all these strategies, the strategies that we can actually use. We also introduce some of the hydrophobic molecules in human serum albumin in order to have more or better loading in the nanoparticles. So we prepare several of the polymers where we introduce the polymer in protein. So it's basically polyprotein conjugates, and then that actually will have the improved power of self-assembly. So we do not need to use the cross-linking agents for this, particularly as we can see that they are basically self-assembling very well. So we also have, as I mentioned also, we combine chemotherapy with photodynamic therapy, and for that we are using fluorine-6 and also platinum drug. Platinum is using as a prodrug, and we also address some of the possibilities of oxygen generation in hypoxic tumors. So that's coming as one of the other advantages that we have introduced in the nanoparticle system. So because of that, we are introducing C6 that as a photosensitizer, and also that PT4, though we are targeting the DNA, and also we are generating the reactive oxygen species, so that actually killing the cancer cells. So there are various other ways that we do that, and we check that whether it is reversing the hypoxia. And we see that, yeah, basically with the biochemical studies, we have proven that our nanoparticle system is actually generating oxygen. So it is very much required, because once the hypoxia activating factor HIF-1 alpha gets upregulated, that actually causes so many changes in the tumor environment, favoring the progression of the tumor. So all these procedures that we are done, so I can't discuss much because of less time. So we also have several graphene oxide systems that I told. So initially we started with the graphite, then it came as like sheets. Then after we put the MPEG PLA on the surface, like amphiphilic polymer on the surface, it turned out to be spherical in nature. So that's beauty of addition of these polymers on the surface of the nanoparticles, the surface of the nanosheets. And we see that it actually reduces the tumor drastically by the photothermal effect. So this is how we see, if we after injection of the system of the formulation, after 4 hours, if we see that and irradiate it, because the biodistribution is high at that point, and if we see that the temperature for our final formulation goes all the way to like 55 degrees centigrade, which is enough to kill the cancer cells. So the cancer did not grow at all by these treatments. So which is highly targeted approach, and it can be explored in the clinical setting as well. So in conclusion, I can say that it's not done yet. So we still have a lot more to do, and so many nanoparticle systems have been tried in the lab. So many we are trying. We are getting better day by day. And our ultimate goal is to develop something which will drastically reduce the tumor volume and will make the tumor disappear, and which is not difficult with the photothermal and combination of chemo photodynamic approach. We can do that. So yeah, so we are enjoying the work that we do. So it's not about the destination, it's about the journey. So with that, I acknowledge the lab members, the funding agencies, ICMR. We had active support from Sun Pharma and Dr. Reddy's lab, ICMR, DST, SCRB, and DBT Biocare. And we have the current research scholars for in the lab, very focused group, but dedicated researchers, past research scholars who worked in some of the projects that I have shown you. And these are my national and international collaborators we work with. And we always look for energetic students in our department to work on all this, because all this work have been done by the research scholars in the lab. So yeah.
Yes.
So now I open the floor for the questions.
Thank you very much, ma'am. It was a wonderful session, and it's a lot to learn from this session. And we actually appreciate all your attempts to solve this untreatable disease called cancer. You have attempted many approaches, photodynamic therapy, chemotherapy, combination of this, and also have tried for various ways to target the cancers. So I request participants, if they have any questions, they can post in chat box. By the time we have some questions, ma'am, from our end.
Sure.
Yeah. So you perform various in vitro and in vivo studies. We would like to know what are the challenges that you face when you done the in vivo studies for testing your nanomedicines for the treatment of cancer.
Okay, so yeah, that's a good question. So we have to first see that which in vivo tumor model we want to develop. So that depends on drugs and how they are used in the clinic. So based on that, then we decide on. So usually some of the tumors develop very quickly, and but some of the tumors take time, and because of the immune response they don't develop also. So sometimes we face that kind of challenges. But then we also, another thing, there is the animal to animal variation. So we don't get the tumor developed to all the animals. So that also happens, but ultimately we always go to n equal to 5 or 6, and then after the tumor develops we give the time, and when it comes to 50 millimeter cube size, then we randomly divide them and then use for the further study.
Yeah, thank you, ma'am. You tried different nanomaterials, like from polymerics to inorganic nanomaterials to carbon-based and various targeting agents also. So throughout your long journey of this research, which are the moieties or agents you find which are promising for these particular studies?
Yeah, so that's also good question. Yeah, so as you see that in the clinic, the nanoparticles based drug delivery systems or nanomedicines are not many. So they are in a very primitive stage. You can say that only we have the liposomes based systems and one human serum albumin based systems. So if that is very primitive. But if you see the preclinical studies in research setting, we have got a huge advancement. So every day so many new systems are coming. But then but when you consider about the translation, then the translation abilities are very poor, because it needs to be done a lot of testing, lot of things, and then ultimately it may not work, because sometimes it's just used as a solubilizing agent but not actually improving the efficacy or the survival of the patients. No, now so far what I found that we can actually explore the human serum albumin and some polymeric materials. The advantage of polymeric materials use is basically they are not so expensive, unlike liposomes that you see that it uses a lot of lipids and everything, which is batch to batch variations are there and it's expensive to get. So if we can convert that to polymeric system, which is better option, and also human serum albumin we can further explore. And if you think about what is the highest therapeutic value we are getting, of course we have to go to the inorganic materials. However, we do not know in great detail about the toxic potential, like what it would happen to our body and all, but that needs to be evaluated carefully. But other than that, those are highly therapeutically efficacious.
Right. Thank you very much, ma'am, for sharing these details. There's one more just a technical question that you have developed the targeting moieties. So what are the different techniques to quantify that the material is targeted in vitro or in vivo?
Yeah, for the in vivo, for the in vitro, you can use the cell-based systems, and with the, of course, your nanomaterial needs to have some fluorescence tagging. Otherwise, you won't be able to determine that where they are. And now, if you have that tagging in the nanoparticles, similar tagging in your targeted and non-targeted nanoparticles, then you can treat the cells with those nanoparticles, and then you can do the check the cells under fluorescence microscope or confocal microscope to see that how much they are getting internalized. So of course the targeted one which have the higher internalization, right? So that you can find out by, and also there is a quantitative way of determining that by the flow cytometry. So you can do that. But when it comes to in vivo, you can actually use the real live imaging just by the intravenous administration. Then you can wait for 1 hour, 2 hour, or 4 hour, or 8 hour, or 16 hours, 24 hours, and see that how it is, accumulating to tumor. So you can see that initially it will have accumulation, but after 8 hours, it may go down for the non-targeted formulation, but the targeted one will stay for longer time. So that you can have one of the studies to find out the targeting potential of your nanocarrier.
Right. Thank you very much, ma'am. There is one question in chat box. How we target vitamin receptors on cancer cells? How it is done for other nano drug delivery systems like nanosponges, etc. How we avoid enzymatic cleavage of these prodrugs during in vivo studies?
Okay, so vitamin receptors on cancer cells, of course, you can put the vitamins on the surface, but usually if you put the vitamins on the surface, like there is a biotin, that you can use. Biotin is used for targeting purpose. So there is biotin receptors on the surface of many cancer cells, and you can put the biotin on the surface, and then you can target the tumor. Okay, so a very easy way to use the biotin on the surface is you can conjugate the biotin with the amphiphilic polymer and introduce that into your nanoparticle system. So that can be done. And nanosponges, I guess that if you are using this kind of system, you need to check about this part size and all, because the size should not be big so that it will clog the blood vessels. So that's why the size needs to be less than 200 nanometer size. So that you have to make sure. And enzymatic cleavage of these drugs, pro-drug during in vivo study, if the pro-drugs are assembling into nanoparticle form, then that will actually avoid the enzymatic cleavage. But if you don't have anything amphiphilicity, it won't make the aggregates. So it will roam around like that. But usually they sometimes form the aggregate, but it could be loose aggregate. But I would suggest if you make it amphiphilic or put the hydrophobic ligands in the hydrophilic polymer, then you will get the aggregates, stable aggregates, which will be preventing the prodrug degradation.